Back in Chapter 4, we learned about mitosis. Now we're ready to learn about meiosis. These are both methods of cell reproduction. Mitosis is used to make somatic cells, which somatic cells are the cells of your body. Meiosis is used to make reproductive cells. These are also called gametes, or they can be called germ cells. These are the eggs and sperm. When you do mitosis, you start with one cell, which is called a parent cell, and it has the full set of 46 chromosomes, which is called diploid. So diploid means you have all 46 chromosomes. In meiosis, you start with the same thing. One parent cell that is diploid. It has all 46 chromosomes. In mitosis and meiosis both, you copy the chromosomes. In meiosis, you go one more. You have another event that occurs when you copy the chromosomes, and that is called crossing over. This does not happen in mitosis. After you copy the chromosomes in mitosis, you divide once, and you get two identical diploid daughter cells. In meiosis, you divide twice. And you get 4 unique haploid daughter cells, which haploid means they only have 23 chromosomes, half the number. This haploid aspect of the gametes is important. Chromosomes come in pairs. So a gamete has one member of each pair. Think of chromosomes like shoes. Your shoes come in pairs, a right shoe and a left shoe, and they go together. Your chromosomes are in homologous pairs. You get one from your mom, one from your dad, and they go together. We'll go through an example now of how meiosis happens. So you start off with a parent cell. So here is our parent cell. And we're going to use different colors for the chromosomes from your mom and dad. So let's say that the green one here is the chromosome from your dad. And we'll make a purple one to be the chromosome from your mom. Now, really, you have 23 pairs, but in our example, we're only going to draw one pair. So in real life, you would have 23 pairs like this. In each pair, there's one from your mom, one from your dad. Okay, so here is, if we say this is step 1, here is step 1, our parent cell that is diploid that we start with. Then we copy the chromosomes and cross over. So we copy the chromosomes. When you copy them, you get the X shape. So each chromosome is copied. The crossing over is when they trade parts. So the chromosomes actually break and switch parts with each other. So you can see where that trade occurred there. This is what gives you unique combinations of traits. Let's do an example. Let's suppose that your dad had a chromosome for type A blood and free earlobes, and your mom had a chromosome for type B blood and attached earlobes. So this picture is here to show you the difference between free and attached earlobes. This earlobe, we did free and green. This is a free earlobe. This one is an attached earlobe. So you can see the free earlobe kind of hangs down here, like this, and the attached earlobe just goes right into the side of the head. There's no little flap that hangs down. That's a simple genetic trait. It's easy to trace. So that's why I'm using earlobes. Blood type is also easy to trace. When you do crossing over, then we have here the original one from your dad, attached and free. We have the original from your mom, type A and free, type B and attached. Then in the middle, we have mixtures. So this one will have type A blood, but attached earlobes. And this one will have type B blood, but free earlobes. So we made a new combination. We basically shuffled the genes you got from your two parents. Then meiosis divides twice. So the first time we divide, we separate those homologous pairs. Then each of those cells divides. And we separate the sister chromatids this time. Now here are our 4 gametes. Oops. And you can see that each one carries a unique combination of traits. So this first one is going to have type A blood and free earlobes. The second one will have type A blood and attached earlobes. The third one will have type B blood and free earlobes. And the fourth one will have type B blood and attached earlobes. So each gamete has a unique combination of traits. We did a really simple example using only 2 traits. You have about 30,000 genes that you can switch back and forth. So there's trillions of combinations you can make. This is why siblings have unique traits from each other, even though they come from the same 2 parents. So meiosis is the process you use to make the eggs and sperm, and you get 4 unique haploid cells.